Hello, dear students. So, we are going to discuss about the biosynthesis of cholesterol and synthesis of paracetamol. This is from natural products. We all know about the cholesterol. Cholesterol has got a bad name. Actually, it is very essential for the existence of life. For having good health, cholesterol, the presence of cholesterol is very much required. It is required for the synthesis of vitamins, hormones, the god. If some force exists like that, they act in our body through the hormones, and this hormone is produced from cholesterol. But there are two type of cholesterol. One is LDL, the other one is HDL. It is LDL is low density lipoprotein, HDL is high density lipoprotein, and high density lipoprotein is called good cholesterol, the other one is called bad cholesterol. There is another partner too, is what we call triglyceride, and if there is more amount of triglyceride and low density lipoprotein cholesterol, it is supposed to have high risk of heart attack. So you need to have more amount of high density lipoprotein in your body. That's how they say. So we are talking about the same cholesterol, and basically the cholesterol is a sterol present in all the mammalian tissue, either in free state or in esterified with fatty acids. Mainly present in the brain and the spinal cord. Solid matter of human brain contains 17% of cholesterol, and gallstone it is completely made of cholesterol. Most of the steroid hormones and bile acids are derived from cholesterol, as I said. Main source of cholesterols are fish liver oil, brain, and means like the brain. In Hindi they say beja khana. Spinal cord and cattle. Linolin from wool is the mixture of cholesterol, palmitate, stearate, and oleate. So if we are looking at the structure of cholesterol, this is how it looks like. The cholesterol is having some parts. Actually, this part is what we call nucleus, A, B, C, D. Three benzene rings, A, B, and C, and one pentacarbon rings. So there are totally 4 rings. That's what the nucleus of the cholesterol. And there are 2 angular methyl group, these two. And there is a side chain which is, there are 8 carbon atoms, and there is an OH group at the 3rd position. So somebody will discuss about how do you name the cholesterol like structures. And here the cholesterol is having 27 carbon atoms. So the discovery, what led to this structure, the proposed structure to be discovered. In 1937, it was reported that the acetate is utilized in the novosynthesis of cholesterol. So we are supposed to discuss about the biosynthesis of cholesterol, and the synthesis process will enlighten something about the how the structure is elucidated. So that's what we are discussing. In 1942, it was demonstrated by Leonard Bloch and David Rittenberg that 2 carbon atoms of acetates are incorporated in the synthesis. We are going to see, and the distribution pattern of the acetate carbon atom in the cholesterol was found by R.B. Woodward, and Conard block, Lebaugy, Doban. These are all not required actually for your exam purpose. The complete degradation of cholesterol. That's how actually in organic chemistry, if you wanted to prove so and so is the structure of a particular compound, you will have many strategy. Out of that, one strategy is that molecule once you produce, you have to decompose it, and the fragment of the molecule after decomposition will give an idea about the previous structure. That's what done by the W. Comfort and George Popjak. The result was confirmed by Woodward, Block and Doban. So the acetate coenzyme is very much involved in the synthesis of cholesterol. At this time, the scoline is reported. This time means the time we have discussed in the previous slide, as 30 carbon unsaturated intermediate, an isoprenoid compound which is the precursor of the cholesterol. Squalene, it is an intermediate during this biosynthesis of cholesterol or the normal synthesis of cholesterol. Squalene is derived from 6-hydroxy acid, is a proof that for the incorporation of acetate in the synthesis of cholesterol, because the squalene is very much synthesized from 3 molecules of acetate. So if we look at our body, what are the sources which will provide cholesterol? The very first source, it is the biosynthesis. Biosynthesis happens mainly in the liver, intestine, adrenaline glands and reproductive organs. And the rest 30% comes from our diets. It's diet intake. We have discussed the liver oil, the fish oil, all those. Now, the focus of this discussion is the biosynthesis of cholesterol, and that biosynthesis of cholesterol involve 5 steps. Now the question is, which are those 5 steps? This is very important question as far as the university exams are concerned. So be very careful hereafter. These are the 5 steps. First one is synthesis of MEVALONIC acid, synthesis of isopentenyl pyrophosphate, IPP, that's how it is called, synthesis of phenacyl pyrophosphate, FPP, and the 4th step is just now we discussed, synthesis of squalene, and the last step is synthesis of cholesterol. And 2 points to be noted are the biosynthesis of cholesterols generally takes place in the endoplasmic reticulum of hepatic cells. The enzymes involved in the synthesis are partly located in the endoplasmic reticulum and partly in the cytoplasm. Now let us look into the first step. So this is how it looks like, the entire process. If you wanted to depict in a single slide, that's how it looks. And the first step, step 1. So you could see that this is 1 acetate. So as we have discussed, you could see 2 acetyl coenzymes, 1 and 2, which give acetoacetyl coenzyme, that also through the Claisen condensation. And Claisen and condensation is, this is how the Claisen condensation could be represented. And here one more acetyl coenzyme is involved in the process in order to produce 3-hydroxy 3-methyl glutaryl coenzyme. And here it is through aldol condensation. We know that the aldol condensation, when carboxylic esters containing an alpha hydrogen bond, that's how we call alpha hydrogen atom, are treated with a strong base such as sodium ethoxide, condensation occurs to give beta ketoester Why an enolate anion? That's what we call aldol condensation. We have already learned, and that's how we get this particular 3-hydroxy 3-methyl glutaryl coenzyme. It happens in the presence of HMG coenzyme synthetase. Here also one enzyme is utilized, that is thiolase enzyme, and this 3-hydroxy 3-methyl glutaryl coenzyme A get reacts with HMG coenzyme reductase in presence of NADPH. It means nicotinamide adenine dinucleotide phosphate, and this is the oxidized form, and you get mevalonic acid. That is the stereochemically very specified one, R configuration is there. That's how the first step goes. Here you could recall it again. The 3 molecules of acetyl coenzymes A are used in the buildup of MVA, that is mevalonic acid. The acetyl coenzyme is mainly derived from cytoplasmic oxidation of ethanol by acetyl coenzyme synthesis in the mitochondria. 2 acetyl coenzyme initially undergo Claisen condensation to give acetoacetyl coenzyme. The third coenzyme combines with acetoacetyl coenzyme by a stereospecific aldol condensation to give a branched chain ester called beta hydroxy beta methyl glutaryl coenzyme. So HMG coenzyme, you would have got the abbreviation. Now what happens? The formation of HMG coenzyme is enantioselective. Only one enantiomer is formed. The reaction occurs in the chiral environment of the enzyme. This is what, if man goes for the synthesis of certain biologically very important stereoisomer, you will end up with, man will most of the time end up with a mixture of stereoisomers, whereas the nature goes for producing a particular compound by using an enzyme. It will give specifically a particular enzyme. That's what this point represents. Here also you could see that enzyme catalyzed reduction of thioester group of HMG coenzyme to primary alcohol gives mevalonic acid. We have already discussed about that. Then next we could go for the step 2. Step 2 is the synthesis of isopentenyl pyrophosphate. We call it as IPP. So here you could see mevalonic acid is taken initially, and it is adenotriphenyl phosphate. IPP is active isoprene unit in the biosynthesis of isoprenoid in general. The IPP is formed by the series of phosphorylation followed by decarboxylation, phosphorylated intermediate, and loss of phosphate. You could see it here. So phosphoric ester, and that's how you get IPP. Here in the third step, the carbon dioxide is eliminated. So here adenyl triphenyl phosphate is involved as a enzyme. Phosphate ester formation. So the reactivity of IPP, the final product we got over here, is such that it can condense into units of 15 to 30 carbons without need of additional ATP. So without using any enzyme, the advantage of this isopentenyl pyrophosphate is it can decompose into it can condense into the units of 15 to 30 carbons without the need of additional ATP. So we will get into the third step. There the important point is the synthesis of farnesyl pyrophosphate. That is the last product over here. So here the IPP is isomerized to dimethyl allyl pyrophosphate, because the double bond should not be at the terminal position. And next what happens is IPP condense with the DMAPP to form geranyl pyrophosphate. That's what we call GPP. It goes with the condensation. Here the terminal methyl group of IPP act as the nucleophile and pyrophosphate of DMAPP as the leaving group. So that's how the condensation happens. So you got the geranyl pyrophosphate, and now this GPP, and the previous slide we have seen IPP, here also we have seen previous reaction. GPP, one more molecule of IPP will add it to the GPP and you get farnesyl pyrophosphate. That's what we call FPP, farnesyl pyrophosphate. So we have seen 3 steps already. What are those 3 steps? Step number 1, synthesis of mevalonic acid. So there are many enzymes involved. And step 2, it is synthesis of ISOPNTENYL pyrophosphate. And we have seen step 3, that is farnesyl pyrophosphate. And the next one, it is the GPP undergoes similar condensation with IPP to give farnesyl pyrophosphate, and which has 15 carbon atoms. You can count how many carbon atoms are there. Now there is a small mistake in the heading, that is step 4, it is synthesis of squalene. So we have taken FPP, and the FPP is written in such a way that it would be easy to understand. So I have accommodated it just like how cholesterol is going to draw, how the cholesterol structure is drawn. And 2 molecules of FPP condense in tail to tail manner to give dihydrosqualene, which is reduced to squalene. So this is what they meant, the tail to tail. So you get dihydrosqualene, and the squalene, what will happen? You don't have to do anything, which will get reduced to give squalene, which has 30 carbon atom. The reaction involves the reduction with NADPH, followed by the elimination of pyrophosphate radicals. NADPH means the previous nicotinamide adenine dinucleotide phosphate, the same. It's an enzyme. And the last step, there it is written step 3 only, this is the 5th step. The last step, it is the conversion of squalene into lanesterol. So this is step 5. Actually step 5 has got 2 parts. One is the preparation of lanesterol, and this lanesterol will get converted to cholesterol. That's how it happens. So you must read NADPH is involved, and you get initially squalene 2,3 epoxide. Squalene has a structure resembles steroid nucleus very closely. Recall that 5-membered nucleus and determine the terminal groups. Squalene has a structure resembles the steroid nucleus very closely. It is converted into squalene 2,3 epoxide. This is what the just upper side, right side, you can see 2,3 epoxide. The enzyme squalene epoxidase causes the second carbon oxygen bond to rupture, which initiate a series of ring closure lead to the tetracyclic lanesterol. So at this point you can see that you have almost reached very close to the structure of cholesterol. So the four members already formed. So here hydroxylase, the enzymes are involved. So you already got lanosterol, and from hereafter the lanosterol will get converted to cholesterol, and it has got two different methods. One is the old way and the other one is new way. Let us see both. Before closure takes place, the methyl group, closure means the resemblance that cholesterol is having 5 rings. So those 5 rings are getting formed in lanosterol, and there how does it happens is, like if you look at the squalene 2,3 epoxide, it's not closed. They are not closed rings. They are written just like closed rings, but they are not connected. So that's what the closure we talk about over here. So before the closure takes place, the methyl group on 14th carbon is transferred into 13th and 8th carbon to 14th carbon. 8th carbon get hydroxylated. So once this lanosterol is formed, it has to get converted to cholesterol, and the steps of the conversion of lanosterol to cholesterol contain NADPH and O2. In the first step, helps in the oxidation of methyl group to removal of carbon. Then mutase move the alkene group. NADH helps in the reduction of ketones. This is how it will be. So this is how it is the old fashioned, like everywhere you could see that lanosterol will get converted to zymosterol through the involvement of bioenzymes, and that will get converted to demosterol, and that will get converted to cholesterol. So the conversion of lanosterol to zymosterol and then to demosterol and finally gives cholesterol. And recently it is reported like this: the most recent studies indicate the pathway involves a easy reduction of 24th carbon double bond to give 24-25 dihydrolanesterol, followed by loss of 2 carbon atoms, and a double bond shift to give 5,7 cholestadine, 3 beta ol, and finally to cholesterol. So you have to, the numbering is very important. You should have a clear cut idea of what is the structure of cholesterol and how the cholesterol is numbered. So I am giving you an assignment. How do you draw the structure of cholesterol? Number 1. And once you draw the structure of cholesterol, number the carbon atom present in the cholesterol. Okay, it's something like nomenclature of how do you name cholesterol as per IUPAC. So you have to draw the structure of cholesterol, number all the carbon atom on the cholesterol, then you have to give the name of IUPAC name of cholesterol. Assignment number 1. So you have already synthesized cholesterol, and you have one more in class question, that is I have means the what are the enzymes involved in the biosynthesis of cholesterol. Okay, what are the enzymes involved in the biosynthesis of cholesterol, and what are their duties, where they are utilized. Okay, I haven't mentioned clearly what are they. You can, everywhere it is available, you can search and you have to list it out. Okay, 2 questions, in class questions. Now one more part left out, it is synthesis of paracetamol. Paracetamol we have heard of, it is analgesics and it is antipyretic. In order to reduce the body temperature, we use it the same way we use it to get relief from the pain. Synthesis of paracetamol, and it is an antipyretic, analgesic, the drug that lowers the body temperature by acting on the heat regulating center in hypothalamus. So paracetamol, that's how it is called. Actually its name is acetaminophen. It could be called 4-hydroxyacetanyl, it could be called N-acetyl paraminophenol. What is the IUPAC name? It is N4-hydroxyphenyl ethanamide. Then the synthesis is very easy. We have to go for nitration in the first step. Then we have already discussed the the reduction of nitro group into amine group. You have already written the oxidation number changes. So you will convert the nitro group to amino group, and the amino group will go for N ester. So you get paracetamol. That's how. This is what one of the important synthesis. If they ask any synthesis of paracetamol, you can write this. So let us just see what are the properties of paracetamol. White crystalline powder, sparingly soluble in water, used in the arthritic and rheumatic condition linked with musculoskeletal pain, headache, neuralgias, myalgias, and dysmenorrhea. It is metabolized primarily in liver, where the major metabolites include inactive sulfate and glucamide conjugates, which are excreted by kidney. Only a small amount of this is metabolized by hepatic cytochrome P450, which is responsible for toxic due to a minor alkylating metabolite. That's NAPQI, means N-acetyl P-benzoquinone imine. The demerit of this paracetamol is a higher dose of NAPQI reacts with liver cell, causes necrosis. This is what about the required information. So biosynthesis of cholesterol is very important. It is not only this much. Like if you write down all these steps, how does it happens? You can't get complete information. Just like if you go for a chemical synthesis, how does it happen? Here the mechanism is not required. You write down what are the reactants involved and what is the coenzyme involved. 5 steps, more than important. First step, second step, third step, fourth step, and fifth step. There are 2 parts. This is how it is. Thank you for listening to me.